DNA, one of the most powerful molecules in the human body, directing the course of life generation. Just in the past few decades, the science industry has harnessed DNA as well as technology to create a scientific field known as genomics. The categorizing and prototyping of DNA has allowed for breakthroughs in generative conditions, as well as a platform for designer DNA. One method in creating DNA is polymerase chain reaction, also known as PCR. This method makes millions of DNA copies from initial template of interest. The application of PCR is widespread, and its production of DNA can result in cloning, mutation detection, as well as sequencing. However, the key to these three applications is successful duplication, which means a test is conducted to see if the intended base pairs are in the DNA segment. Because number of base pairs is directly correlated to length, technology has been developed to separate and categorize DNA based on this parameter. One of the most commonly used methods is gel electrophoresis. Because of DNA's phosphate group, the molecule is negatively charged. Thus, when an electric current is applied to DNA in the presence of a membrane, diffusion occurs. This happens specifically in a membrane known as agarose. A derivative of seaweed, this membrane takes gelatinous form for separation during electrophoresis. Essentially, the gel will form a sieve-like matrix and work by suspending molecules of DNA within the gel as it diffuses. This will result in a larger molecule having a shorter distance of diffusion than shorter ones. Taking a look at the process, protocol can be divided into three sections: gel and DNA preparation, equipment setup, and testing. To create the gel, agarose is added to a buffer at high temperatures, then placed in a horizontal electrophoresis tray to cool to required firmness. Additionally, DNA is dyed and loaded into wells within the gel. Testing begins when conductive probes are attached to the apparatus, sending a direct current through. This acts as the driving force, causing negatively charged DNA to diffuse out of the wells and begin diffusing across the tray to the positive probe. Once diffusion is complete, the gel is removed from the apparatus and placed under an ultraviolet light for imaging. Test samples are measured next to a marker ladder, which is made of a mixture of DNA of known length to serve as a reference. Overall length is then measured in base pairs as the unit length. On a more microscopic level, to discover how the DNA is diffusing through the gel, we look at agarose as a molecule. Comprised of two monomer subunits, this linear copolymer creates a 3D gel matrix when mixed with a buffer and cooled. The cross-linked matrix forms as agarose bundles aggregate together and keep their shape with hydrogen bonding. Additionally, there are considerations in reference to agarose separated as a membrane. Primarily, the concentration of agarose needs to be considered, because a greater density will cause shorter diffusion lengths. Additionally, the diffusion process of DNA can affect the membrane structure. For instance, when DNA moves through the gel, a buildup phenomena occurs where aggregates begin to drag, creating a less symmetrical matrix. Keeping in mind the properties of agarose gel, there are many mathematical models that attempt to explain the motion of DNA diffusion through the membrane. This is done to better understand what variables affect diffusion, as well as create a tool for researchers to predict theoretical distance diffused before running an experiment. This ensures the correct protocol is used. The models that were analyzed attempt to describe diffusive motion in the application and relate this to DNA length. First, we take a look at the free volume model. Here DNA is represented as a sphere, and the fractional volume for DNA to move through becomes a ratio of the agarose mobility to the interstitial space. Ultimately, the model defines a retardation factor for the gel to describe diffusive motion. For a better representation of the analyte, we look at the reptation model. Here, DNA is described as a snake moving through the gel matrix, where the channels between the pores are represented as a tube. Mathematically, diffusion becomes a function of temperature, friction, and the number of occupied pores. Overall, both models, as well as others, can be combined to create a diffusion profile. This is functional for a wide range of application that will ultimately predict DNA length. As an engineer, it is important to be able to design a process to achieve a variety of desired products depending on need. Although a simple process, this can be applied to gel electrophoresis to analyze a wide variety of DNA lengths. Although standard operating conditions use a range of 0.8 to 1.5% agarose density, this value can be increased for shorter DNA lengths and decreased for longer ones. This accounts for a tighter matrix for smaller molecules of DNA to diffuse the same length as larger DNA molecules in a looser matrix. A summary of all operating conditions can be seen in the referenced table. Keeping agarose concentration constant, the following variables have inverse effects on time and distance. For instance, an increase in applied voltage and tray size result in a quicker diffusion. Looking at temperature, there is a maximum value temperature can reach before it becomes a liquid. However, if the temperature becomes too low, firmness will hinder diffusion time. An optimal value can be found in this range. Overall, agarose gel electrophoresis has been the dominant separating technique in DNA application. Along with the models discussed, computer software is able to describe diffusion and relate this to length. Not only accurate, but also versatile and simple, gel electrophoresis provides efficient separation and analysis in the molecular chemical industry.